Darby will be speaking on demonstrating complex generic product bioequivalence, benefits, and considerations when using new analytical methods. Welcome, Darby.
As Markham mentioned, I'm Darby Kozak. I'm the Deputy Director of OGD's Division of Therapeutic Performance 1. In this session, I'll give an overview of FDA's perspective and a few examples of recent research on new analytical methods for demonstrating complex generic product equivalence. My talk will cover 3 areas of complex drug product development where new analytical methods may be beneficial. These include methods to characterize complex active and inactive ingredient properties, methods to characterize the physicochemical properties of complex formulations and dosage forms, and finally, the use of new tools and methods to characterize the performance and bioequivalence of these complex products. For each area, I'll give an overview of the perceived analytical need, then some recent GDUFA-funded research outcomes, and finally provide a few examples of recent complex products that may warrant research and use of new analytical methods. In the past years, GDUFA-funded research on new analytical methods have generally covered a broad range of products and research priorities. For this session, I will focus predominantly on the research done on developing methods to characterize complex active ingredients and formulations, and on new tools being developed for evaluating product equivalence. For complex active and inactive ingredient characterization, there's a general need for sensitive analytical methods that can quantitatively characterize the chemical structure of heterogeneous macromolecules, such as natural and synthetic polymers. In addition, there are also instances where high-resolution methods may be needed to characterize the impurity profile down to trace amounts for complex actives. Some recent GDUFA research is focused on using solid-state NMR, LC-MS, and GPC with 4 detectors to characterize these complex ingredients. For example, a GDUFA grant to Dr. Eric Munson via the National Institute for Pharmaceutical Technology and Education examined the use of solid-state NMR to characterize the polymer structure of butyrimer, a complex cross-linked synthetic polymer composed of 3 different monomers. Given that this polymer is not readily soluble in most solvents, conventional solution-based NMR is limited. The solid-state NMR method developed was able to accurately determine the ratio of each monomer and was used to evaluate lot-to-lot variability of the reference listed drug. These findings not only better inform FDA reviewers on the RLD product, but also aim to provide generic industry with a method to develop and compare their proposed generic to the RLD. Similarly, a number of GDUFA research projects have examined new analytical methods to characterize the properties of polymer excipients. A large portion of these projects have focused on methods to reverse engineer and characterize PLGA, which is a biodegradable polymer commonly used to enhance drug release in long-acting injectables and implant products. These include methods to separate and characterize differences in the polymer molecular weight, L to G ratio, in-group chemistry, and even characterize linear versus branched polymers. This is important as this comparative testing of the proposed generic polymer to the reference to the drug polymer can be used to support formulation sameness, as well as these properties have been shown to impact product performance, including the drug release rates. Of recent interest are analytical methods to characterize complex oligonucleotide products. Particularly challenging are developing and assessing methods that can characterize the oligo sequence and any chemical modifications to both single and double-stranded oligo products. Moreover, these methods will likely need to be very high resolution to characterize and compare the impurity profiles of the proposed generic and RLD product to assess potential genotoxic risks. 2 recent oligo products approved include viltolericin and lumiricin, a single and double-stranded nucleotide with chemical modifications to the backbone, in-group, and even base pairs. Internal GDUFA research projects have been initiated on these complex oligo products. These projects utilize samples of the RLD product to develop methods that can characterize the key aspects of these complex actives. This includes characterizing process-related impurities, such as insertion or deletion of a base pair, and how those changes impact the oligo sequence and relative frequency that a specific impurity sequence may occur. In addition, there's need to characterize process and degradation-related impurities resulting from modifications of the oligo base pairs and the backbone chemistry. Although research projects have been initiated, more research and testing is likely needed to facilitate the development and approval of generics. For complex formulations, there's a general need for analytical methods that can measure and compare the critical quality attributes of formulations. Demonstrating comparable physical-chemical, or Q3 properties, helps address and limit potential modes that a product may not be bioequivalent. Some recent GDUFA research is focused on imaging methods that incorporate spectroscopic analysis to provide a more informative and quantitative analysis. These can be used to differentiate chemical regions within the formulation and local distribution post-administration. An example of this is the use of focused ion beam scanning electron microscopy combined with imaging analysis or other chemical-specific analysis such as energy dispersive X-ray spectroscopy. This combination enables cross-sectioning of a solid material via ablation using the focused ion beam and image profiling the structural and chemical-rich regions. In this example, which is the outcomes of a recent GDUFA contract to GM Solutions, the polymer, active ingredient, and void regions of a PLGA microsphere were characterized. This method has the potential to understand how changes in the manufacturing process may impact the microstructure of the product, which may lead to a potentially inequivalent product performance. Similarly, GDUFA research projects conducted by Professor Richard Guy at the University of Bath and Professor Connor Evans at the Massachusetts General Hospital have been exploring the use of confocal Raman microscopy to better understand how formulation properties or differences in a generic formulation can affect the cutaneous penetration and thus local pharmacokinetics. This can be valuable to supporting bioequivalence of a locally acting product such as topical dermatological drug products. Recently approved complex formulations that may warrant additional research and development of new analytical methods to support equivalence are the ocular implant Durista and the ultrasound contrast agent XM foam. Durista is a PLGA intracameral implant for extended release of bimatoprost to reduce the interocular pressure. Methods are likely needed to characterize the PLGA properties as well as the drug release and degradation rates. XM is unique in that it is the size and structure of the foam that gives rise to the ultrasound contrast signal. Therefore, methods to measure these properties of the foam as well as response to ultrasound signal may be explored to ensure generic product equivalence. The last category of new analytical methods I will highlight are the development of tools to assess product performance and bioequivalence. In general, these tools are intended to more efficiently discriminate between bioequivalent and non-bioequivalent products. In addition, these tools can help bridge knowledge gaps of how the critical quality attributes of a product can impact the in vivo performance and therefore may be used to support alternative bioequivalence approaches. Recent GDUFA research has focused on developing more sensitive and discriminatory in vitro release testing methods, as well as tools that may be more accurate mimic the variability of human physiology and product use conditions, which may be also used to support equivalence. An example of a newly developed tool to assess product performance is the electroanalytical in vitro release method created by researchers at the U.S. Food and Drug Administration's Jefferson Laboratory Nanotechnology Core Facility. This unique method enables the continuous and direct quantitation of doxorubicin released from liposomal formulations, and it overcomes limitations and inaccuracies of conventional separation and analysis methods. This method may provide a more sensitive evaluation of potential formulation and manufacturing differences that may give rise to a non-equivalent product. Other GDUFA research projects have focused on developing new tools that better reflect the variability of human physiology and product use conditions that can help bridge in vitro to in vivo performance properties. This includes a number of GDUFA-funded projects to develop more realistic mouth-throat models and inhalation profiles that can provide a more realistic evaluation of lung deposition of drug particles from orally inhaled drug products. These new tools can help demonstrate how changes in the patient's physiology may impact the performance of these products, and moreover, may be a more sensitive measure to compare to conventional imaging and sizing methods of how differences in the delivery device can affect the generated spray properties that may lead to differences in drug delivery. Ultimately, these tools and tests may help bridge potential knowledge gaps that would otherwise need to be addressed by a comparative clinical or pharmacokinetic study. In summary, I hope that I've highlighted how GDUFA research on new analytical methods have improved characterization of complex active and inactive ingredients, complex formulation evaluation, and creating new performance and BE assessment tools. That this research aims to aid both generic industry and FDA reviewers in the development and assessment of complex drug products, that this research is continually needed as new analytical methods and complex products are being developed, and that feedback is needed to focus GDUFA research in areas that will have the greatest relevance and impact to industry and the public. Finally, I'd like to acknowledge a few of the internal and external collaborators who have worked on projects I presented in this talk.
All right. Welcome back. So, folks on the panel, please turn on your videos, and we can start sharing. Fantastic. And again, we have largely the same panel as our last one, and this time we get to comment on Darby's talk. So I'll start with, does anyone have any preliminary thoughts about Darby's talk and maybe a relationship to Rosario's talk? And maybe we could start with Rosario's thoughts about Darby's talk, if that's okay with you, Rosario.
Yeah, sure. Phenomenal talk, Darby. Really enjoyed it. Good overview of some of the newer techniques and the way that they're being used, for sure. And I just go back to what was said before. Is the technique is one thing, but access to the technique and the sample preparation piece. Would love to see more around sample preparation, and to me that's really where a lot of the variability comes as well too. Not necessarily all the time from the instrument, but those are some initial thoughts. Love to hear others as well. Thank you.
Sure, if I can just add in there, just to reiterate, Darby, really great talk. But I think the last slides about the amount of research and the usefulness of that research can only be reiterated. How excellent it is to have the FDA doing this work and supporting all the industry, whether it's generic or even the specialty companies, to get the work there. This is really greatly appreciated by us, that the money's being spent this way. It really is helpful, and it's the variety of techniques, the pushing the thoughts that we've got to keep in mind as well, how clinically relevant some of the differences are. I mean, I take the inhalation product, the development of several different throats. Which throat do you use, which is going to show you the clinical relevance of the difference between your devices. The one that shows you the least difference, or the one that shows you the greatest difference, and how does that reflect your clinical result? And that's constantly in the mind of the industry's thinking, and this work, your work that you do, really helps us to get to understand, first of all, some of the science better, but also the FDA's thinking on it. So it is greatly appreciated all the way.
Thank you, Kevin.
Thank you.
Darby, would you like to respond to Rosario or Kevin? And then we can go to some of our other panelists.
Sure. No, I think, like I said, I think it's all a collaboration amongst all of us. Do you mean without understanding what's really the industry's need and some of the new ideas that are coming out there? I mean, FDA can start to perceive some of those things, but really kind of getting that feedback is a key component. And what I've seen through a few talks today in particular that we'll probably get into a bit more is liposomal formulations, the characterization, what types of techniques be done there. And what I've seen an awful lot again too is the oligonucleotides, a really exciting new sort of field, a lot of different sort of analytical challenges because of the complexity of that molecule and the potential unknowns associated with it. So I think there's going to be a lot of really good sort of focus into those components, as well as some of the other sort of the bridging sort of modalities, these in vitro sort of innovative sort of tests that can help bridge that what comes out of the drug product to its in vivo performance. You mean that sort of that gap between there, those mouth throat models that you mentioned, Kevin? Do you mean is it more representative? Is this going to help us to better understand that inherent variability and not be able to necessarily be detected by a spray plume geometry? This will then give us a little bit more confidence in the fact that how those subtle differences may actually impact the overall performance in the range of potential patients. Do you mean those types of bridging, I think, is the key things that we want to better understand?
So one of the questions that we promised our participants that we'd address is what research is needed to prepare for generic versions of oligonucleotide drug products, the siRNA, chemically modified antisense oligonucleotides, etc. I think both Rosario and you, Darby, have both mentioned some plausible technologies that could be used. I wanted to hear from some of our other panelists whether they agree with some of the highlights that Darby and Rosario brought out, or are there other things that you would think about that we would need to prepare ourselves for looking at generic versions of oligonucleotide drug products regarding bioanalytical methods? Any volunteers to address that?
Yeah.
Go ahead, Malshar.
Markham, this is Maleshwar. Yeah, so thank you for that talk. Very interesting work on the automated in vitro release testing and the oligonucleotide characterization. What will greatly help in characterization, standardization for the oligonucleotide is that the way we evolved on the peptide products, several characteristics using multiple orthogonal techniques, and publishing those articles, really how to use those techniques and how to interpret them and how to characterize the generic product in comparison with reference products. So those dimensions will significantly.
You're an advocate for publishing our research, and we agree. There's doing the research and not communicating it is just like not doing the research at all. You've got to, if FDA is doing research, we will communicate it. I understand that from industry, when you do research, you may or may not publish it. What can you identify some of the barriers that you have in industry regarding publishing scientific work that gets towards understanding some of the gap of funded research from your industry to looking at suitable test procedures and techniques that could be more widely adopted, and so that FDA and others, I think there was a push, some industry folks said, oh, regulatory agencies need to recognize these methods. But if you're doing the research and you're showing that you're getting good results, but you're not able to publish, then that's not getting it out there. So I wanted to hear from industry what some barriers you might have encountered. And you don't have to speak specific for your company. Maybe you heard from another company. So maybe Dama, Rosario, or Ravi, or Kevin, any thoughts on that?
Yeah, Markham, I think.
Yes, we
am
hear
I
you.
audible, Markham? Thank you. So I think so. the research what we do it is so when we work, so we work in a very competitive environment. So the most challenging stuff is like so when we publish it, sometimes competitors will take an advantage of it and then take those efforts. And this would be the research is an efforts of years together, and then when we publish, obviously it goes to the public domain. And so where company is spending significant amount of money, that is where we have any vision to publish this work. Otherwise, the industries are doing a fantastic work, no doubt in that, and that is where we are coming up to the agency in the form of scientific discussions and then discussing all that. So that is one of the inhibition to publish the research work what industry is doing currently.
I guess it depends on the novelty and the challenges that you might encounter. For example, some research you can bring into FDA at our pre-ANDA meeting, and we would have sufficient expertise to decide, yes, this is a valid method, or we might critique the method and say, hey, that method has known problems out there. But if it's sufficiently novel that there's very little knowledge at FDA or even around the world on it, then that becomes a problem. And I think that's when you have novel methods that need further communication and as they're being developed. Pahala?
Markham, I think so. With respect to novelty of the method, there is no issue. So when we are coming to the agency, absolutely fine. There is no issue at all. When we wanted to publish this work, so which is going to the public access, then there is a concern from a competitive advantage. Mm-hmm. And even sometimes it could be a simple particle size method. So probably how do you look at the particle size? Looking at an orthogonality, so altogether it gives a different dimension. For example, you take the microparticles, which is the PLGA based microparticles. The data what you get it from light scattering, the laser diffraction, you get it something, and then when you look at the microscope, is altogether it gives a different information. And how do you understand the true particle size? So even these two together we can publish a research article. Mm-hmm. What is the right tool to find out the particle size of a microparticles? But important thing is when we can't publish this to the public access. So obviously when we come to the agency, so we are giving it and we are discussing and then getting the feedback from the agency.
Pahala, Darby, are we keeping ahead of the curve enough, do you think, to be ready for industry when they submit proposals?
I don't know the answer to that, Markham, but I think it's a matter of having a good communication, just like some folks have mentioned through like Panda meetings type, to have that dialogue to share the information and we go from there. But one thing I would like to ask Darby, I'm just curious out of curiosity. There are a few projects ongoing. They're all good stuff. Where are we with respect to the project? Midpoint or almost at the end of some of these projects? They're very interesting.
I could just say another word around this. I think it's super important. The product development meetings, in my opinion, have been super useful. A lot of great information exchange. As you know, right, there's long lead times. There's long lead times to those meetings. So the next point would be could we use the control correspondence route if you have a novel method or you have a question around the sample preparation to engage with the FDA on, instead of waiting to put the whole body of data together, in that product development package. Would love to hear from the FDA on that point. Thank you.
Darby, did you want to take that one?
Yeah, I can. To quickly go on to Pahala's, I mean, a lot of the projects are all at different sort of stages. A good component there, especially publicly facing, is the GDUFA public website. It gives us an annual report, so you'll kind of see what the status is of new projects and publications in that sort of realm. So that's always a good sort of resource. You can also look at the RFAs and things like that are being generated as a point in which you can then see what new projects are being initiated and sort of the direction and focus that are coming out from this discussion here. So then to get back to Rosario's, I think that's a really interesting proposal. I hadn't seen, I have not necessarily, and I definitely solicit Ram and Pahala's sort of feedback on this, where we've had such a type of control correspondence. It might be an aspect where what we call right now the 120-day or more complex control correspondence can come through, but I need to look at that in more detail on how the correspondence route has been taken for some of the things that you're proposing here. I personally have not seen that previously, but I know that those conversations are had a lot more in detail in the pre-ANDA space, because usually we're talking about then it's data driven and content driven, and allows us to then have a more substantial discussion back and forth. But we can probably get back to you, or find something a bit more on that. But I can't say anything on the control.
I just wanted to bring up the issue of scientific questions addressed to the agency. The complexity of it varies from one issue versus another. And from the FDA standpoint, when we get a question and it involves, say, multiple divisions, multiple teams looking at their question and responding to it, say the analytical method question impacts both bioequivalence and equality, then more than one reviewer is needed to that. And so it does take some time for them to both look at it and then meet and agree on the response to industry and factor in what other things they know about that method and other products that have tried to use that method. So those things add a little bit more time to those kinds of things. The more novel something is, the more time we might have to take to either research it or come up with finding out who has expertise on it so we can answer it in an appropriate manner. So I'm going to ask our audience here to make sure they throw any questions that they might have for our panel, either for this session or our wrap-up panel session that's coming up after our next talk. So think about questions that you might have
Can you hear me
that are
now?
relevant to the topic at hand, and go ahead and type those into
perfect. Thanks.
our
So I just
comments
have
section,
a request
so we can
for
then
the agencies.
take those and
we have seen a lot
think
of
about those and answer them
research around the
in the
IVRT,
session.
IVPT
So
works on
this is
the
an invitation to all of our
semi-solid products and
participants.
everything, right?
Right now we
there
have
was a limited research and
over
publications
200 some
around the
participants.
ophthalmic product,
So please
IVRT
go ahead
works.
and
So can we see more
we
research
would welcome any questions
public,
that you might have.
in the
Right,
public
panelists?
domains or for public or industries, that how does
Is
this
there folks that I haven't heard from? We haven't heard from. Robbie, are you able to get your microphone thing straightened out? Excellent. You're coming through loud and clear. Go ahead. Absolutely. I think we're going to start with the dermal products, but we can move on to other products going forward. You heard from other speakers at this meeting, and I'm going to encourage all of our audience here to go ahead and, when you see, go to the Center for Complex Generics website, and you'll see for August the 18th, 19th, and 20th, I believe, are the 3 dates that we have an IVRT, IVPT workshop, where we're going to be discussing all of the techniques, the receptor solutions, the
the apparatuses. Actually, I've learned that there are actually multiple types of apparatuses that you can use, and what is the correct way to use it, which apparatuses are preferable over others, and perhaps which ones are most appropriate for use for certain products. And there's room to look.
I think first of all, as Ravi brings up a great point,
ophthalmologics as well, at least to start considering those as you listen to the experts in the world of IVPT and IVRT. So a plug for the August meeting that's going to be run by the center. Other speakers have given a plug as well, and so I thought I would do my part here. You all should definitely go to that site and look it up. The site is up
one of those potential bridges,
and
there needs to be a lot of innovation there. I think you mentioned there in the dermal space, they definitely have an IVRT, IVPT.
Darby, you had a question. Go ahead. And I think you wanted to ask a question of the panel as well. Please feel free to do so.
I think some of the challenges for some of the other dosage forms, the emulsions, the suspensions, and things like that is how do you do an accurate release, especially something that may have burst release or something that have a very rapid release. How do you accurately measure those? Or things that have a very slow release and being able to do that and then be able to size separate. So I've seen where like field flow fractionation's been used. I think Rosario, you even brought up a nanotechnology. I think it's multiple filaments. Those types of things are really interesting. I think what we need to do is as a group explore those more, better understand how they can be employed, what type of information they give us, what are their limitations, and then be able to then apply those in a concerted manner to be able to say, this is the problem it's addressing, and here's its limitations. When I run underneath these conditions, this system doesn't work anymore. Underneath these conditions, we can see a very good release, and this is able to tell small differences between our formulations, and therefore we help it feels it gives us that bridge to better understand that these products will be bioequivalent. So to me, I think this is a prime area for a lot of good research, and there's a lot of new analysis methods and techniques out there compared to the conventional either membrane or a dialysis bag. I mean, those things have such low surface area that it's challenging.